Hello, BNG312 class, and welcome to this lecture on microbiology. This is supposed to be an in-class lecture, of course. Hope you're all enjoying the snow day. So let's begin. But first, just to let you know that there will be an online class on Monday. That's February 13th. Basically, you will be learning about Newtonian and non-Newtonian fluids. I briefly mentioned these things in Wednesdays and Mondays lectures. You'll learn a little bit more about them, and perhaps we'll put them into a biomanufacturing example. So we'll just have to see. So there won't be any physical class on Monday the 13th. And the assignment that's associated with this online class will be due on Monday the 20th, the usual time. Okay, so we talk about microbes and bacteria, yeast, other microscopic organisms. They're everywhere. They can be found in great numbers in the soil. They can be found in the oceans. They can be found in great numbers on us, as well as in us, and on other animals and in other animals. In fact, there are a lot of efforts these days to characterize the types of bacteria, yeast, etc. that are present on our skin, in our bodies, as well as on and in other animals, because there could be the potential for some interesting biotechnological advances when we do this. So in fact, microbes are so ubiquitous to have truly microbe-free conditions. It doesn't happen very often. You may think it does, but it doesn't. They require a conscious effort. So when you're performing surgery, like some of you probably will after med school, or if you're in the biomanufacturing business, you want a relatively germ-free environment. And this takes some doing: sterilization, disinfection, and keeping that environment microbe-free, aka germ-free. But we won't get into that today. So let's start with bacteria. What do bacteria do? Well, what do bacteria do that benefits bacteria? Well, they grow, they divide by binary fission, so they split apart, they copy their DNA and they split apart, and then they respond to stimuli. And we'll see some examples of this. But why do we care what bacteria do? Well, you probably have, if you watch the news and you listen to me talk in class, you probably know some of that. First, bacteria can cause disease. They can cause disease in plants and also certainly in animals. You've heard of E. coli, you've heard of botulism, you've heard of anthrax. These are bacterial diseases, or E. coli causes many types of disease, or can cause. And we'll get into that in a little bit later. We care about bacteria for nutrition because bacteria can actually metabolize and provide compounds for animals and for plants. The bacteria that live in us can provide us with vitamins. They can break down some starches and things like that we eat and produce necessary metabolites that we can absorb into our blood that keep us healthy. They can also do this for other animals. Some animals will eat wood and grasses. Bacteria can digest those celluloses. Bacteria can decompose dead organic matter. In fact, if bacteria didn't do this, we would be piled sky high with a bunch of dead things because they would never decompose. But because bacteria and even yeast do their work, these dead things go back into the soil. Bacteria also make valuable compounds. We've found bacteria that can make acetic acid, acetone, butanol, ethanol, bioplastics, etc. We've talked about some fungi that make acetic acid, and we've talked about some fungi that make ethanol. But bacteria can do these things also. There's bioremediation. So bioremediation is the detoxification of compounds. There's some bacteria that are really good at breaking down compounds and making them less toxic. If you were with me in BNG319, we talked about some bacteria that can degrade polychlorinated biphenyls. And bacteria can do many other things as well. There's a lot of things that bacteria can do. And here are some interesting ones. There's a bacterium called Shigella onidensis, which was found in Lake Oneida in New York. It's a facultative anaerobic bacterium, more about what that means in a bit, that can detoxify heavy metals and can conduct electricity. Ralstonia eutropha is a bacterium I care about a lot. It is a bacterium that can perform bioremediation. Some species can degrade those polychlorinated biphenyls, and also produces a bio-based biodegradable plastic. And you can see inside these bacteria here, you see little white lumps or white dots. And those are called granules that are full of this biodegradable plastic. Deinococcus radiodurans is an interesting one. It's the most radiation-resistant organism known. The mechanism of resistance comes from the fact that it's also desiccation resistant. So you can really dry it out as well. But that also gives it some radiation resistant properties. It has over 8 copies of its chromosome, so there are 8 copies of its DNA in its cell at all times. So if the DNA gets damaged, it can use the other copies to repair. And then there's this Alcanivorax borkumensis bacterium. It's a bacterium that's found in the ocean. And you see here behind an oil spill. Well, this bacterium will nucleate, or it'll start to collect around pools of hydrocarbons, pools of oil, like oil spills. And it will consume that hydrocarbon. So this basically is an oil eater. And they have been known to be found, along with other oil-eating bacteria, they've been known to be found in great quantities after an oil spill. And so scientists have characterized areas that have had oil spills, like in Alaska in the 80s. And of course, some of you may remember the oil platform in the Gulf of Mexico that blew up, that spilled out a bunch of oil. Well, there were a bunch of bacteria there that were eating that oil. Okay, and I'm going to show you a little video. I'm not sure how this is going to work recorded, but I'll show you a little video on a really interesting bacterium. Just interesting as far as the novelty of it all. It may not necessarily be interesting biotechnologically, but this is a bacterium known as Bacterial vibrio that can attack, burrow its way into, and eat E. coli bacteria. So here we go.
Before we start the movie, we see a tiny black Vibrio predatory bacterial cell inside a bigger, fluorescently labelled prey E. coli cell. The Vibrio has invaded this cell, and as we start the movie, we see the Vibrio elongate bidirectionally, growing and using the insides of the prey cell. Then it septates unusually to give 5 progeny cells, and the E. coli goes dark as those progeny cells lyse it and escape and go on to attack more prey bacteria.
Okay, so that was Bacterial vibrio. Well, so bacteria, like I said, they're everywhere. They can be found in all sorts of different environments. They are that diverse. And microbes that are found in certain areas have special names. So if we are looking at bacteria that are found in the cold, those would be called psychrophiles. The optimal growth temperature for those, it would be less than 20 degrees Celsius. So these are bacteria that can potentially grow in the refrigerator if they wanted to. Mesophiles are what we're most used to. These have optimal growth temperature anywhere from 20 degrees to 50 degrees Celsius. So anything that's found associated with our bodies would be in that category, as well as most things that are found associated with soil would grow optimally in this temperature range. Thermophiles are special and are very interesting in terms of biotechnology, because their optimal growth temperature is greater than 50 degrees, so they can withstand heat, which means their enzymes and things can withstand heat. And over here you see Thermus aquaticus, a bacterium whose DNA polymerase is well known. It's used by molecular biologists all over the world. You know it as Taq DNA polymerase, which is used in PCR. Halophiles will grow optimally in the presence of salt. They can grow in these bacterial mats in these very hypersaline environments. They can be used to make things like fermented foods. So they can be used to make things like sauerkraut. And they can remove pollutants from salty wastewater. So they have a lot of value there. Acidophiles grow optimally in acidic conditions. So if you can imagine, they grow in pH of less than 2. Alkalophiles can grow optimally in alkali conditions, so high pH conditions. Okay, unfortunately we can't do the Socrative right now, but anyway, I'll just go through it. We are examining bacteria as a source of protease enzymes to add to laundry detergents. Say we are formulating a Super Duty laundry detergent that is most effective around the boiling point of water, so around 100 degrees C. Then what type of organism should be the source of our protease? Well, the answer to that is, because of that high temperature, we want thermophilic bacteria, because the protease enzyme should be able to withstand the temperatures around 100 degrees C. That would have been answer C on Socrative. And one of the ways that we can characterize bacteria is by whether they grow in the presence of oxygen, whether they need oxygen, or whether they will only grow in the absence of oxygen. And these are called aerobes and anaerobes. And there are several shades of aerobic and anaerobic activity. So you can have bacteria that are strict aerobes, meaning they will only grow in the presence of oxygen, like Pseudomonas, for example. So if you have a tube full of some thick media and you inoculate with some bacterial cells, a strict aerobe is only going to grow at the surface, near where the air and the oxygen in the air can actually penetrate. They're not going to grow down here where the environment is essentially anaerobic. You can have obligate anaerobes or strict anaerobes. Things like Clostridium species fit the bill here. And again, if we had a tube of thick media here, you inoculate it with bacteria, you would only see the anaerobes growing down here in the bottom, where the environment is essentially anaerobic. But a lot of bacteria, like E. coli, for example, are what are known as facultative aerobes or facultative anaerobes, meaning they can grow in an aerobic environment or they can grow in an anaerobic environment. So they're pretty versatile, and E. coli certainly is pretty versatile. There are also microaerophiles, meaning they will grow in the presence of oxygen, but they don't want too much. So if you have a tube of media, you're not going to see a collection of microaerophilic bacteria at the top, like with strict aerobes. You'll see a collection of microaerophilic bacteria a little ways down the tube, where the oxygen can't penetrate a lot, but can penetrate at least a little bit to provide that microaerophilic environment. And then there are aerotolerant organisms. These are actually organisms that will grow in anaerobic conditions, that grow best in anaerobic conditions, but they can also survive in the presence of oxygen, or can even grow in the presence of oxygen. A good example is the colon bacterium Bacteroides fragilis. And bacteria come in many shapes and sizes, and these are excellent feats of engineering, these bacterial designs here. They optimize things that they can do, and the things that they can do well. But typically the size ranges from about 0.5 micrometers in length to 50 micrometers in length. And they have an ideal surface to mass ratio for absorbing nutrients and gases and such. So you see a lot of them look like a pill shape, or look like, a curved sausage type deal, or maybe even as spheres or elongated spheres like spheroids, as well as these spirillum that look like corkscrews, or these spirochetes. There are exceptions to this. This bacterium Thiomargarita is a rather large one, at perhaps even up to 300 micrometers in length. But again, this is a highly specialized bacterium and is not found in that many places. So they can come in a wide variety of shapes and sizes, and this all influences how they gather nutrients, how they swim through fluids. So these spirillum and these spirochetes are known to just corkscrew their way through fluids, and they can do a lot of different things. because they have a variety of different parts here. We have the flagellum, which allows bacteria to swim. We have capsule, which is kind of a polysaccharide layer that covers a bacterium. In bacterium that cause disease, these bacteria have capsule to evade the immune system. And in fact, a lot of bacteria have capsule to prevent them from being eaten by things like amoebae and other predators that might be there either in the soil or in an animal. Bacteria have a cell wall which keeps them in the proper shape. They have a plasma membrane, again, which is a barrier for most things, unless it's something they want to take up. And then, of course, there's the cytoplasm, which has things like ribosomes, plasmids, potentially. We'll talk a little bit about plasmids. And has the nucleoid, or the circular DNA, or the chromosome here, tightly packed inside the cytoplasm. Okay, so here is a picture of a bacterium that has been broken apart, an E. coli bacterium. You can picture that it was like right here at one point. It's been broken apart, and its chromosome is just spilled out. So the DNA in an E. coli bacterium, or any bacterium, is very tightly packed in there, in that cytoplasm. So you may wonder, there's not a whole lot of room for anything else. So E. coli basically have to control everything and regulate everything to the extreme in order to survive and thrive, because their cytoplasm leaves no room for error. So bacteria usually contain one circular chromosome that contains all their genetic information. They have genes that are needed to grow and divide. And you should note that the chromosome isn't always circular. In the case of E. coli, it is circular. Sometimes there are more than one distinct chromosomes. Again, it depends on the bacteria. There's a lot of bacterial diversity. So generally, chromosomes, like in bacteria, are circular. And if you stretch one out completely and take a picture of it, like this intact electron micrograph here of a circular bacterial chromosome, you see that it looks like a big loop. However, eukaryotic chromosomes, that is, eukaryotes, cells that have a nucleus in them, have these linear DNAs that are wound and tightly packed around these proteins called histones, and they are compacted into these chromosomes, which you may recognize if you've ever looked at karyotypes of your chromosomes or of other mammalian chromosomes. They kind of look like these big X's or Y's even. So they're quite different. However, the bacterial chromosome is packed inside a small space. These chromosomes in eukaryotes are packed around histones. And again, both require some strict regulation in order to access and express some of the DNA inside. So again, the bacteria don't have a nucleus per se, the way eukaryotes do. But that is an advantage if you're a bacterium, because bacteria can couple transcription and translation, which means they can produce RNAs and then they can produce enzymes all in one space, whereas eukaryotes have to shuttle RNAs outside the nucleus where they get expressed in places like the Golgi apparatus and the endoplasmic reticulum. Here's a quick look at cell division. Again, bacteria divide by binary fission. They replicate their chromosome, then they begin to split apart until there are two cells with two separate chromosomes. It's a little more complicated in eukaryotes. We have mitosis, which is the general splitting apart of the cells. Then we have meiosis, which is what happens in our germ lines, where we're producing 4 cells, each with half the chromosomes that we originally had. We have 4 in the beginning, we have 2 here. And then they recombine in some manner. If you're a yeast, you do yeast mating, and you produce a new diploid organism. So you have 2 copies of chromosomes in the eukaryotic cell, but you only have 1 copy at the end of meiosis. And we mentioned briefly about plasmids. Well, plasmids are circular typically. Sometimes they're not, but usually they're circular pieces of DNA They're what is known as extrachromosomal. They're not part of the bacterial chromosome. They're not necessary for day-to-day survival of bacteria. They do, however, confer certain advantages to bacteria that have them under some conditions. So that's why plasmids persist in the environment for sure. And we can use them as bioengineers. We can manipulate them. We can put genes on them. We can keep plasmids in bacteria for the sake of biotechnology and biomanufacturing. In the wild, how do cells get plasmids? Well, they can get them a number of ways. They can get them by conjugation, whereas a cell with a plasmid can mate in a way, or can conjugate with a bacterium that doesn't have a plasmid, and the cell with the plasmid can replicate the plasmid and also transfer it to the recipient cell here, until eventually both cells, the host and the recipient, or the donor and the recipient, have a copy, an intact copy of the plasmid there. We can also do it in the lab via transformation, where we have a bacterium that can grow only in the absence of antibiotics, but we basically can kind of shock the cell membrane open a little bit, and very briefly, so that we can then get a plasmid inside. And once that plasmid is inside the bacteria, we can plate on a agar plate that has antibiotics on it, and then find any resistant bacteria. And we can be sure that resistant bacteria are containing the plasmid that we want them to contain. Okay, so there are various advantages to having a plasmid if you're a bacterium. One, and probably the most popular one, is antibiotic resistance. Plasmids can sometimes confer the ability to kill other organisms. They have what are known as bacteriocins on them, which are bactericidal compounds that are made. They have the ability to infect a host. They could confer that. They can confer the ability to persist in a host also. They can also confer the ability to utilize a different carbon source. In one case, there's a plasmid that is found in some bacteria that gives them the ability to use carbon dioxide as a carbon source. Antibiotic resistance, however, is the most often talked about, and we most often take advantage of it in genetic manipulations, especially if we are trying to create organisms in biotechnology. So basically what we would do is take a plasmid, we can engineer it, we can cut it with these things called restriction enzymes. And again, if you had me in BNG312, we talked about this a little bit. And then you can put a gene into a plasmid, ligate it together, put that plasmid in a new bacterium, and the bacteria will make a certain product because they have the gene with which to express the necessary machinery to make that product. And in this case here, you see this little flow chart. These bacteria are making what is known as recombinant insulin. Okay, so again, I'll just go quickly through the Socrative question. We want to develop an E. coli strain that produces recombinant insulin. We have a plasmid that has the insulin genes on it, and we can insert that into E. coli cells, which we do. How can we tell if the E. coli cells have the plasmid containing the insulin genes? Well, the answer to that is we can test for antibiotic resistance. The plasmid should carry, if we're good bioengineers, we should have given them a plasmid that has an antibiotic resistance marker on it, which would allow the bacteria containing that plasmid to grow on a certain antibiotic. So we can test that, and we can assume, at least initially, that all bacteria that can grow in the presence of an antibiotic have the plasmid that can allow E. coli to produce insulin. Okay, so we can compare quickly before we move on to talking about eukaryotes for a bit. Compare bacteria with or prokaryotes with eukaryotes. So, you can see there's some differences in the number of chromosomes. Usually bacteria only have one. Usually eukaryotes have way more than that. DNA, is it found in an organelle for bacteria? No. Eukaryotes, yes. Is DNA compacted into chromosomes? Bacteria have histone-like proteins, but it's not really the same. It's basically just compacted into a small space known as the cell cytoplasm. Eukaryotes, they are compacted into chromosomes. There's no nuclear membrane in bacteria, and there's the formation of a partial diploid in bacteria, and I haven't really gone into that. But you can add some genes in duplicate or even triplicate if you wanted to in bacteria, but you don't have to add every gene back in triplicate. This doesn't necessarily happen in the eukaryotes. There aren't many organelles in prokaryotes. There are photosynthetic apparatus in photosynthetic prokaryotes, as well as in photosynthetic eukaryotes. Flagella are fairly simple in prokaryotes. They're much more complex in eukaryotes. Both have mechanisms to store carbon and other nutrients. Bacteria can store it as bioplastic. They can store it as starch. They can store it as lipids. Eukaryotes can do the same thing. Plant cells can store starch, and our adipocytes can store lipids. Spores, some bacteria can sporulate. They do what's called endospores. The spores are created inside the cytoplasm. Eukaryotes can have either endo or exospores, spores that can form outside the cytoplasm. Bacteria are relatively highly heat resistant. A lot of that has to do with cell walls. Eukaryotes not as much, certainly not animal cells. So eukaryotes. Eukaryotes have an entirely different cell parts list. Although they do have things like they have DNA, they have proteins, they have ribosomes, things like that. They have these fancy structures known as organelles. Nucleus where the DNA is stored, things like the endoplasmic reticulum where proteins go to be fully matured. Golgi apparatus, same thing happens on the way to being secreted. If you're a plant cell, you have chloroplast where the photosynthesis happens. Peroxisomes get rid of waste products, misfolded proteins and such. The mitochondrium produces the ATP in the cell. So bacteria can do what yeast and a lot of other eukaryotes can do. That's for sure. Except one of the main differences in eukaryotes, everything has its own compartment. So photosynthesis has its own compartment. Building ATP has its own compartment. You could think of it as a department, I suppose. And transcription has its own compartment as well. Yeast, which are eukaryotic cells, can divide by budding. So it's not true binary fission necessarily, but you have kind of a small bleb or cell forming on one pole of the organism, where eventually you get some nuclear material migrating in, you get mitosis happening, and then finally the bud grows to be bigger and bigger, and then it breaks off. You get what is called cytokinesis. And suddenly you have two yeast cells, but one is clearly a mother cell and the other is clearly a daughter cell. And that daughter cell grows, and then the budding process starts all over again. So what do yeast do? Well, yeast can do the same sorts of things as bacteria. They can grow, they divide, and they respond to stimuli. What roles do they play? Well, they can do things like alcohol production, ethanol. We know Saccharomyces cerevisiae is good at that. They can even produce things like lipids. They can also produce things like other what we call commodity compounds. And of course, we went over with Aspergillus niger can produce a lot of citric acid. So that's one of the things yeast can do. They decompose plant matter, animal matter. They cause disease as well. Oral thrush is just one example of a disease that can be caused by a yeast, plus much more stuff. There are other non-yeast eukaryotic organisms, protozoa, as well as some other types of organisms that are found in the environment, found in fresh water and stuff. Diatoms are among them. Diatoms are handy because they have those hard shells that have a bunch of good surface area. So might have heard of diatomaceous earth that can be used as a filter aid. Well, it's basically made up of a bunch of dead diatoms. And you can see they have fairly exquisite looking shells. But these shells can aid in the filtering of wastewaters, pool water, and things like that. Okay, so we said that both yeast and bacteria can grow. Certainly they can grow in the environment. We want to make them grow in a fermenter culture to produce value-added products, as I like to call them. So all organisms require an array of nutrients in order to survive. They need, just like we need, the essential elements. So they need carbon, we're all carbon-based life forms here, oxygen, nitrogen, phosphorus, sulfur, magnesium, hydrogen, as well as some others. Metals and other compounds that we need in a very small amount. Actual organic compounds that we need, or that microbes need, again depending on the organism, amino acids, nucleotides, lipids, vitamins, and cofactors, but most organisms, or many organisms, can make their own. E. coli can certainly make pretty much most of its own amino acids, nucleotides, lipids, and vitamins and cofactors. It rarely has to get whole cofactors from the outside environment. And so what do they need most? Well, carbon, they need a lot. Microbes need around 10 to the negative 2 moles per liter, which is rather a lot, relatively speaking. The next most, they need nitrogen, around 10 to the negative 3 moles per liter of culture. But they also need other things like sulfur, phosphorus, potassium, magnesium. Oxygen, essentially, is ubiquitous, and as long as some is dissolving into your culture broth, you're doing all right if you have an aerobic organism. If you have an anaerobic organism, you have to get oxygen from some other way. Okay, here's a short list, a very abbreviated list, if you're growing organisms in biotechnology, of some carbon sources and nitrogen sources that are used in industrial fermentations. So things like starch, molasses, even like alkanes for bacteria that can eat up oil, cellulose, waste, so things that are left over from dead grasses and other plant matters. For nitrogen source, you can use things like soybean meal, which contains a lot of protein. Yeast extract also contains a lot of protein. And a lot of these things have one common thread in that they contain a lot of protein. You can also use many more different things. For nitrogen sources, you can use just pure urea or ammonia. Carbon source, you can just use refined glucose. Depends on what you want to do. But in a lot of these cases, you're using waste products and extracts because you are trying to reduce the cost of microbial fermentations. Okay, so another Socrative question we'll go over real quick. You are attempting to grow some bacterial biocatalysts in a fermenter. You add molasses to a volume of sterile water, inoculate with bacteria, and close the lid and walk away and let the fermentation begin. But the next day you come back and the bacteria haven't grown. Well, what went wrong? Well, one of the main things I can think of that might have gone wrong is that you did not add sufficient nitrogen. It could also be that you're missing some other important nutrients in there as well. So you need molasses as the main carbon source, but you could also add like a yeast extract, distillers solubles, something like that as the nitrogen source. And that would probably also give you other nutrients as well. Okay, so talking of nutrients, bacterial cells and other types of cells move towards food and also can move away from things that are what we call environmental insults or poisons or toxins. So this is called chemotaxis, where bacteria, yeast, amoebae will run towards a food source. So let's look at that with an amoeba doing this. So there's a pipette tip, and you can see they're moving that pipette tip around. because that contains some food for the amoeba to eat. And the amoeba really wants to get at that food. You can see it's moving towards, it's extending those little appendages, moving towards that food to try to get at that tip there. But this is chemotaxis, and a lot of bacteria can do this. Okay, so we can look at rich media, which contains sources of carbon, nitrogen, sulfur, phosphorus, etc. And is often produced from things that are digested, like enzyme-digested peptides, lysed and dried up cells, ground up meat even, etc. It's basically fairly crudely prepared broth that has all the possible nutrients and stuff that bacteria would like to eat. It is what we call an undefined media. So we kind of have an idea of what's in it, but we don't know exactly what's in it and how much of given nutrients are in it. Defined minimal media is media that we make for bacteria to grow that contains certain amounts of things that we add individually. So it contains all these things like carbon, nitrogen, sulfur, phosphorus, etc. It is also buffered, so it can withstand pH changes, which means that bacteria can grow to a higher level, to a greater density, because their waste products are being buffered, so they're not going to die of acidification. So we don't have to add much else unless our microbial nutritional requirements dictate. So we can add vitamins, cofactors, things like that if we need to. All of this is sterilized, certainly with rich media is sterilized, so that only our bacteria of choice grow. In many industrial fermentations, however, defined minimal media is used. And for that reason, it is defined so we can get repeatable results, so we don't have much variation. And you remember I said a while back, variation in product annoys the customers. Because nutrient limitation can have a role to play in industrial fermentations and in production, we can define that nutrient limitation into our media. So for example, if we want to make some xanthan gum, we can have the media set up so that we run out of nitrogen at a certain point. Same with PHA, polyhydroxyl alkanoate bioplastic production. We can run out of nitrogen, we can run out of phosphate, for example. And that will trigger the production of these products. And you can imagine there are some differences between microbial growth in rich media versus minimal media. If microbes are in rich media, well, they have everything they need. They can just pick it up from the outside. So they can grow a little bit faster, as can be seen with this E. coli culture that's growing in this rich media, this blue line here. But if they're in minimal media, they grow, and they may eventually reach the amount of cell mass that they can reach in rich media, but it's going to take them a lot longer, because a lot of the stuff they need to make, they have to actually make, like a lot of amino acids, nucleotides, and things like that. They can't just pick them up from the media, and that takes time. Okay, so we've been talking about E. coli. Well, you've probably heard in the news at some point that E. coli caused disease, and you're probably thinking at some point that, well, E. coli are bad. Why are we using them so much? Why do we try to make insulin with E. coli? Sounds dangerous, doesn't it? Because E. coli has been linked to many food poisoning outbreaks from tainted meat, dirty vegetables, but we talk about it, I talk about it quite a bit, as using it in biotechnology and biomanufacturing. How is this even possible? Are these the exact same E. coli? The disease causers and the biomanufacturers? Not the exact same. So the disease causers are pathogenic E. coli, like enteropathogenic E. coli, uropathogenic E. coli. These contain special genes that encode for what we call virulence factors that allow bacteria to basically cause disease. And a lot of these E. coli can have things that will allow them to lyse red blood cells, that will allow them to infiltrate, say, cells that are present in our guts. And that is the result of the special DNA that happens to be in these E. coli. However, we have this E. coli strain K12, the most commonly used lab strain. This doesn't have these pathogenic DNA bits in them, or what we call pathogenicity islands, and I'll show you what that means in a little bit. Because they've been grown in the lab forever and ever and ever, and essentially they just either, if they ever had the pathogenicity island in the first place, they've lost it. And the result is that these E. coli, these K12, they can't even grow in the human gut. So I could scoop a glob of E. coli K12 off a plate and eat it, and I would be fine. It might taste awful, but I would be fine. I wouldn't get sick. So how do E. coli go bad? How do they get these pathogenicity islands that make them cause disease? So they can pick them up from the environment, which they do. They can just pick up DNA lying around from lysed cells. And if that DNA happens to be a pathogenicity island, then, well, they incorporate it into their own DNA. They can cause disease. Or they can be infected by a bacteriophage, a virus that spews bacteria into the E. coli cell, which can then recombine and get put into the bacterial chromosome. Or they can pick up a plasmid from somewhere. They can gather these from different environments, different plasmids. They can pick it up, and they can somehow recombine into the E. coli genome. And then suddenly they're enteropathogenic, uropathogenic, enterohemolytic bacteria that can cause pretty major disease that can potentially kill folks. So what do I mean when I say pathogenicity islands? I talk about DNA islands, like islands of DNA that are separate from all the other genes in the chromosome that have the genes that cause disease. And they have genes that allow these pieces of DNA to be put in the bacterial chromosome. And they're essentially foreign DNA, and we can tell if we look at what we call the GC content of bacteria. So in this case, E. coli. We can look at the GC content, and that means a certain amount of guanine and cytosine present in the DNA of our bacteria. So there's overall, if we look at an E. coli chromosome, there's a certain percentage. And it's not actually 50%, it's around 30%, 37% or something like that. But a pathogenicity island, if we just look at that, it has a different GC content. Maybe it's got a lower GC content than our E. coli genome. Or maybe it's got a higher GC content. So we can tell that, hey, this is a big stretch of foreign DNA. And that foreign DNA contains all the virulence genes. And so, again, the E. coli strains that are used in industry don't have these islands, these pathogenicity islands, nor are they going to get them from anywhere. We're using them in isolation. We're trying to keep them in a fairly germ-free environment to avoid our E. coli from going rogue. Okay, so essentially, in summary, bacteria exist and grow in numerous environments, almost anywhere, but they do need available water. But there's water most places. They can eat pretty much anything, even other bacteria. Bacterial cell structures are different from eukaryotic cell structures. Eukaryotes have organelles, whereas bacteria don't have organelles per se. Bacterial structure is different from eukaryotic genetic structure. The chromosomes, very different. Both are compacted in certain ways, which feeds into how they regulate their gene expression. And bacteria and yeast can play many different roles, both in the environment, in nature, in our bodies, and in biotechnology. Okay, so for further reading, if you have the yellow Schuler-Kargi text, you can read Chapter 2, which is a little survey of microorganisms and how they might relate to what we do in biotechnology. And hopefully I will see you on Wednesday. Monday, again, we have a online classroom activity, and I'll post that on Monday. If you have questions for me, please feel free to send me an email at any time this weekend or next week.